Um, Hello, everyone. I'm really excited to present our research with the whole microbiome community across the globe. So let's get started. So for today's presentation, I'm going to be talking about the altered mucosal IgA response to oral and fecal microbiota in patients with axial spondyloarthritis. Now, spondyloarthritis, it's an umbrella term used for multiple diseases such as axial spondyloarthritis, acute anterior uveitis, psoriatic arthritis, and inflammatory bowel disease. And this group of disorders has a common genetic factor association, which is human leukocyte antigen B27 or HLA-B27. In addition to this common genetic factor, these diverse diseases also have common clinical features. These could be articular in nature, involving enthesitis, axial skeletal inflammation, peripheral arthritis, dactylitis, or abnormal bone formation. There can also be extra-articular manifestations, which could be inflammation of the eye, inflammation of the gut, as in IBD, and inflammation of the skin, as in psoriasis. So, before we go into the role of microbiome, why do we even suspect gut microbiota in spondyloarthritis? Well, because of the overlap or the presence of gut inflammation in spondyloarthritis. And as it's been shown for the last decade or more that gut inflammation is associated with microbial dysbiosis. When we look into patients with axial spondyloarthritis, around 50 to 70% of those patients will have subclinical gut inflammation, or they'll have microscopic gut lesions. And of those, 6 to 10% patients will go on and develop overt IBD. On the other hand, 50 to 80% of IBD patients will have arthritis. Mostly it'll be peripheral arthritis, but then in some cases, around 10 to 20%, it'll be involvement of axial arthritis. The other piece of evidence comes from the rodent models, which have been shown to be highly dependent upon the gut microbiota. If we take the example of HLA-B27 transgenic rat model, the disease development is dependent upon the gut microbiota, as HLA-B27 transgenic rats reared in a germ-free environment will not develop either inflammation or arthritis. However, if you add back even normal luminal microbiota or even altered Schedler's flora, there will be development of arthritis and gut inflammation. In addition, another model, the SKG mice, the presence of certain types of microbiota will increase the incidence as well as the severity of arthritis as well as gut inflammation. So with this slide, I just wanted to kind of bring different flavors of microbial dysbiosis, if you will, that have been associated with spondyloarthritis. We have a general microbial dysbiosis where studies have reported alterations in the microbial community structure or the alpha diversity. And we also have seen papers where they've shown specific microbes as the disease markers for spondyloarthritis. For example, there's one paper by Prevotellaceae and colleagues where they've shown Ruminococcaceae navis as the disease marker, and another study has shown dialister as the microbial marker. However, this is not the case for all the microbiome studies. Some studies could not find microbial dysbiosis associated with spondyloarthritis. It could be either absence of a difference in microbial composition or lack of differences in alpha diversity. In addition, research from our group on HLA-B27 transgenic rats has also shown that microbes, which may have the highest changes in the relative abundance, may actually not correlate with host immune dysregulation and therefore may not be drivers of the disease and could be actually changing in response to the gut inflammation. Sorry, my slides moved. So what is the immunological relation of microbiome with the disease. So this is the direction that we've been going. And in this experiment, what we have done is we have taken patients who have axial spondyloarthritis in healthy individuals, and we've taken samples for stool and saliva, and we have done microbiome analysis or 16S sequencing. In addition, to understand the immunological effect or relationship with the host, we have also performed IgA sequencing. In previous studies done by Noah Palm and group, they have shown that microbes that are targeted or coated with IgA have cholitogenic nature. So when they transferred IgA positive microbes into germ-free mice, those mice developed colitis. However, when they transferred IgA negative microbiota into mice, those mice did not develop colitis. So in this experiment, we wanted to understand if there is differences in the IgA coated or the microbes that have immunogenic potential in patients with AxSpA. So we sorted them based on their IgA coding and then performed 16S sequencing. So here I'm showing you the IgA coding analysis from different fractions. So first panel is the fecal samples, and then is the saliva samples. And row one, so here are healthy individuals, and these are patients with axial spondyloarthritis. And if I could bring your attention to this panel specifically, where we are looking into the patients with axial spondyloarthritis, we can appreciate that number of microbes are actually enriched in their IgA coding. And we look into the microbes that constitute this group, we find that many belong to the family Ruminococcaceae or Lachnospiraceae, which have been shown in previous studies to be associated with inflammation. And we did not see, interestingly, increased IgA coding in saliva samples. We went on further to perform disease correlations to understand if there is relationship between the IgA coding as well as the disease activity index. And again, we were able to find a microbe belonging to the family Ruminococcaceae in the fecal samples of patients with axial spondyloarthritis, as well as 2 species from the genus Proteobacteria in the saliva of the samples. However, in all these cases, the correlation is inverse. So these microbes are actually decreased in their IgA coding in axial spondyloarthritis patients as compared to the healthy controls. Going on further, we also wanted to see if the IgA coded microbes have a different metabolic potential as compared to the IgA negative microbes. So we went ahead and performed computational analysis using the software PyCrust-2 to infer metabolic pathways. And here are the fecal metabolic pathways from an IgA positive fraction, and on the bottom panel is the IgA negative fraction. And you can appreciate that in the fecal samples, we observed an increased relative abundance of multiple pathways which belong to the to inflammatory pathways as well as metabolic pathways belonging to amino acid biosynthesis in the fecal samples. However, in contrast, in the saliva sample, most of the pathways were actually decreased in patients with axial spondyloarthritis. Now, it's interesting to note that despite the larger percentage of microbes being uncoated or IgA negative in the gut, we did not see enough changes in the metabolic potential, kind of bringing us back to the immunogenic role of these IgA-coated microbes, which needs further investigation. So to summarize, I'd just like to, I hope that I have shown you data that suggests that IgA sequence in axial spondyloarthritis patient has revealed enrichment of several fecal microbes. And we also observed decreased IgA coding in both fecal and salivary microbes that correlated with disease severity. And the inferred metagenomic analysis suggests that there's perturbation of inflammatory and metabolic pathways in IgA positive microbial compartment of axial spondyloarthritis patients. In future, we are looking into the metabolic profiling of these microbes, and we would like to look into the actual role of these immunoreactive microbes to understand how IgA is maintaining the microbial community structure as well as axial spondyloarthritis pathogenesis. And with this, I'd like to thank James Rosenbaum, my mentor and collaborator, and my other collaborators, Mark, Lisa, and Tammy from OHSU, as well as members of the Rosenbaum Gill Lab. And here are my funding sources. Thank you so much for listening.